All right, so thanks everybody who's here on time. We'll kick off in just a second. Please welcome Dr. Erin Giddis, whose lab is at Carnegie Mellon University. She did her training, PhD training at UC San Diego, studying the vestibular system in Sasha Deloc's lab. And then following that, went on and did a postdoc with Anatole Kreitzer at the Gladstone Institute at UCSF. This is where she began studying the basal ganglia. And I think you then started your lab at Carnegie Mellon in 2012. Is that correct? So welcome. We're really excited to hear you talk today about the work going on in your lab. And if everyone could hold questions till the end, I'll call on people when they raise their hands and you can get your questions in afterwards. Cheers. Thank you so much.
All right. Thank you, Lauren. And thank you for the invitation to speak here today. And thank you to everybody that's listening. I know that these Zoom seminars are getting tedious, kind of 2 years into the pandemic. So hopefully we can keep this lively and have some time for questions at the end. All right, so the title of my talk is Circuit-Based Strategies for Long-Lasting Motor Recovery Following Dopamine Depletion. And let me see if I can hide my meeting controls so I can actually see my slide titles. There we go. So the focus of this talk is the neural circuitry underlying motor symptoms of Parkinson's disease and how we can kind of use our foundational knowledge of how these circuits work to develop better treatments for the disorder. So Parkinson's disease is the second most common neurological disorder after Alzheimer's disease, and it affects about 10 million people worldwide. It's broadly classified as a movement disorder, and a summary of some of the symptoms are shown here. Some of the most prominent symptoms are a severe decrease in movement and inability to generate voluntary motor commands. The disease is caused by the loss of dopamine neurons in the substantia nigra pars compacta, which are kind of shown here. And the bulk of the dopamine produced by these compacta neurons innervates the basal ganglia, and especially the input nucleus, the striatum. So in Parkinson's disease, as these dopamine neurons are lost, you lose this key neuromodulator from the basal ganglia. And this causes a host of circuit problems within the basal ganglia and its associated structures, many of which are important for regulating movement. And so as a result, the main symptoms that are used to diagnose Parkinson's disease are motor symptoms. All right, so as I just kind of said, dopamine is a critical neuromodulator that helps to keep normal circuits in the basal ganglia functioning. And the basal ganglia sit at this important crossroads of a bunch of different motor systems within the brain. The corticothalamic system, basal ganglia also, especially recently, we're realizing just how important it is for regulation of motor circuits in the brainstem. And normally when dopamine is around, it is kind of like the oil that greases the wheels of the basal ganglia and keeps the output of the basal ganglia very regular. So it's able to kind of coordinate all these different motor commands and keep the gain of your motor output appropriate. When dopamine is lost, this causes the output of the basal ganglia to get highly irregular, and it sends out this noisy signal that kind of overwhelms the smooth transmission of motor commands throughout the brain, and creates this kind of difficulty in initiating and executing movement that we see in the disease. So if we take a look within the circuitry of the basal ganglia, it's rather a complicated system, I think, is once you really take a close look at just about any system in the brain, it starts to get very complicated. And so we need a way to kind of figure out how can we intervene in this system in order to restore function. And one of the keys to discovering this is having an animal model where we can really test out the different parts of the circuitry of the basal ganglia. So the animal model that we use is a 6-hydroxydopamine mouse model. in which we can infuse a toxin called 6-hydroxydopamine into the axon bundle, going from the compacta to the striatum. And when you infuse this toxin, 6-hydroxydopamine, within a couple of days, you can get a near complete depletion of dopamine from the striatum. So this is an immunostain for tyrosine hydroxylase, which normally kind of fills the striatum. So these are all the axons of dopamine neurons innervating the basal ganglia. And within a couple of days of infusing the 6-hydroxydopamine, you can see that dopaminergic innervation is nearly completely lost. This causes a host of cellular and circuit changes within the basal ganglia, which I will talk more about in a couple of minutes. And these changes cause motor symptoms in the mice, many of which resemble motor symptoms seen in human patients, including a severe restriction of movement. You can see in this picture, they also get this kind of stooped posture and they walk with a shuffling gait, a lot like the motor symptoms seen in human patients. And something important for this talk is that one of the physiological changes that we see in the basal ganglia is the output nucleus, the substantia nigra reticulata, changes from this highly regular pattern of firing to this disrupted, bursty pattern of firing. Neurons also start to fire very synchronously together. And this resembles the patterns of physiological changes that you've seen in human patients. So this is a recording of a basal ganglia output neuron in a human who's undergoing surgery to implant a deep brain stimulating electrode. You can see that there's a lot of similarities in how the physiology of mouse neurons change after dopamine depletion compared to what's seen in human Parkinson's patients. So there's a lot of kind of predictive validity from the 6-OHDA mouse model that we can then use to study how neural circuits in the basal ganglia respond to the loss of dopamine, and then how we might be able to intervene and what parts of the circuit do we want to tackle in order to restore motor function. So one of the areas that has been most closely studied in Parkinson's disease is the input structure of the striatum, where it's hypothesized that these two kind of pathways in the basal ganglia originate: the direct pathway, which facilitates movement, and the indirect pathway, which suppresses movement. So the direct pathway is so named because the medium spiny neurons in the striatum that express D1 type dopamine receptors, they project directly to the basal ganglia output nucleus. Their activity is thought to be too low when you deplete dopamine for a variety of reasons. And so the prediction is that if you put channelrhodopsin and use optogenetics to very specifically boost the activity of these cells, that should restore movement in a Parkinsonian mouse. And this experiment was done about 10 years ago in Anatol Kreitzer's lab. I wasn't involved in the study, but I was a postdoc there at the time. And in my lab, we replicated this in our dopamine depletion model because we just wanted to make sure that we could use optogenetics and that we would see similar results. So if you express channelrhodopsin in the direct pathway neurons in the striatum, and you render mice Parkinsonian with 6-hydroxydopamine, these depleted mice spend almost all of their time in the immobile state. But when you stimulate the direct pathway and get that pathway going, you can see that this relieves the immobility. So the mice start moving around the arena, and then you turn the light off and the mice become immobile again. And you turn the light on and you stimulate the direct pathway and the mice start moving around again. You can just kind of do this on and off. We stimulated 10 times in total. And you can see that every time we turn the light on, you see this big increase in locomotion. The mice are free to explore around the arena, but they can only keep moving while the light is on. Once we turn the light off, the mice very rapidly drift back into that immobile state. And this really kind of represents the where we're at currently in terms of treating Parkinson's disease. So there are a number of therapies. Some are pharmacological, some deep brain stimulation, which I'll talk about in a minute, involves electrical stimulation of circuits within the basal ganglia. All of these therapies can be highly effective at restoring movement in Parkinson's patients, but as soon as the deep brain stimulator is turned off or your drug dose wears off, the symptoms come right back. So we haven't found ways to kind of persistently restore movement in Parkinson's patients or in animal models of Parkinson's disease. So this kind of is a major challenge, and is what we need, the next step that we need to take in treating this disease. So current therapies are not ideal for long-term care because they lose efficacy or cause side effects over time. And this means that we need new strategies that can repair, not simply mask brain dysfunction in Parkinson's disease. And these clinical challenges can be met by basic science because basic research asks how neural circuits in the brain are organized and how they function. And this work will lead to new insights into which neural circuits are the best to target in order to maximize therapeutic benefit while minimizing side effects. And I would say kind of an ultimate goal for treating not just Parkinson's disease, but all neurological disorders is, can we sort of marry insights from basic research to clinical practice in order to find ways to induce therapeutic plasticity? So how can we guide or reinforce healthy patterns of brain activity in order to repair the function of damaged neural circuits? So I'm not sure that we're all the way there yet, but I want to share with you some work that my lab has been doing in order to try to get a better sense of ways that we can intervene in the basal ganglia. This is based on insights that we've gained about the neural circuitry there, and that it's already kind of yielding better therapeutic benefits than what we currently have. All right, so I've already shown you how stimulating the direct pathway can lead to acute relief of motor symptoms, but the effects don't persist. So within about 30 minutes after stimulation, immobility is basically back to where it was before treatment. But I haven't shown you any work that we've done intervening in this kind of other half of the basal ganglia in the indirect pathway. This is the pathway that's thought to be kind of overactive in Parkinson's disease. And because the function of this circuit is to oppose movement, it seems like a very good, place to target. This pathway, the indirect pathway, is also thought to be one of the main sources of that irregular pathological kind of oscillating bursting activity that is a physiological hallmark of the Parkinsonian state. And a key nucleus within this pathway is the external globus pallidus, or GPE. If you do the circuit math, it's thought to be underactive in Parkinson's disease. So the output of both of these types of striatal neurons is inhibitory. So the GPE is overly inhibited in the Parkinsonian state. And this is backed up by recordings in across all animal models of this disorder, as well as recordings in human patients. So the kind of first place that we started to see if optogenetic interventions in the indirect pathway could overcome these motor deficits in dopamine depleted mice was we put channelrhodopsin in the GPE to try to boost its activity. And surprisingly, we found that this did not work. So when we globally expressed channelrhodopsin in all the GPE neurons and went ahead and stimulated, there was maybe a little bit of movement that we saw in the mice, but it was nothing very impressive. And again, this was surprising because the GPE is really kind of the only output nucleus for these D2 neurons in the striatum. And so based on what we knew about the basal ganglia circuitry, that this should have worked. So why didn't it work? Well, one of the assumptions that we made in that experiment is that all neurons in the GPE are the same, and that is clearly not true. There are a bunch of different kinds of neurons in the GPE, and I think the field hasn't yet decided exactly how to best target these different populations of cells. But we have some transgenic mouse lines that give us some tools in order to start picking apart some of these populations. And one of the major type of neuron in the GPE, or sort of the canonical GPE neuron, express the protein Parvalbumin, or PV. And so we wondered if we went back and we did our experiment and this time restricted channel rhodopsin expression just to this Parvalbumin expressing subset of GPE neurons, would we get a different therapeutic effect? So by being a little more selective and targeted in where we're expressing channel rhodopsin, can we get a better therapeutic outcome? So we redid this experiment, this time just expressing channel rhodopsin in the Parvalbumin expressing GPE neurons. And sure enough, this time we saw a dramatic restoration of movement in our Parkinsonian mice. The gray line up here are mice that are expressing UIFP. So there's no active opsin being expressed in these mice. You can see that they stay immobile throughout the entire experiment. But in mice that have channel rhodopsin expressed in their Parvalbumin expressing GPE neurons, you can see that there's a little bit of movement when we turn the light on, but the much stronger effect is this sort of gradual increase in mobility over the course of the experiment, such that by the end of the experiment, the mice, even though we're not stimulating anymore, the mice are still moving around in the open field. This is about a 10-minute effect, which is nice, but nothing to get too excited about. But when we kind of redid the experiment and left the mice in the open field for much longer, you can see that the mice remained highly mobile in the open field for the duration of the experiment. We let this experiment go for 4 hours. So the duration of this therapeutic effect is really striking, especially compared to the direct pathway, stimulation of the direct pathway, which I showed you at the beginning. They yield about the same amount of kind of transient effect, but the therapeutic effects of direct pathway stimulation decay very rapidly compared to those of cell type specific stimulation within the GPE. To the right here, I'm just showing a kind of a gross summary of what the mice are doing as they're moving around in the open field. Compared to a dopamine intact naive mouse that we kind of plop in the open field for a bit of time, if we take a Parkinsonian mouse and we restore its behavior, you can see that the types of behaviors that they express are kind of similar to what we see in naive healthy mice. They spend about the same amount of time walking, grooming, doing fine movements. The one thing that we don't recover is rearing. So our rescued mice still don't rear like healthy mice do. But this kind of is meant to show that they can stop and they can switch behaviors, so they're not just sort of pathologically circling around the arena. So this was a really exciting result, but it was also a little puzzling. What was it that we were doing with our cell type specific stimulation that we weren't doing when we were globally activating all of the GPE? So in both cases, these red Parvalbumin neurons are being stimulated. So why does it matter if they're stimulated by themselves versus with everything else in the GPE as well? So to address this question, we wanted to record locally within the GPE while we were applying our optogenetic stimulation. So we integrated an optical fiber into our recording array. These mice are awake and head fixed on a rotating wheel. And we find that when channel rhodopsin is globally expressed in the GPE, when we turn the light on, we turn on all of the GPE neurons. So channel rhodopsin is working as expected. So now what happens when channel rhodopsin is just expressed in the red neurons, right? It's very easy to kind of fall into this sense when you're doing an optogenetic experiment that you're just manipulating the cells that have the channel rhodopsin. And you kind of forget that these cells are all integrated into larger networks within the brain. So even if your channel rhodopsin expression is specific, you can sometimes have effects on you probably always are having effects on multiple circuits, even those you're not thinking of. And that was kind of what was going on here. So we repeat these experiments, we're recording from the GPE while we're stimulating. And if we kind of do an optotagging protocol first to try to estimate which cells are directly expressing channel rhodopsin, therefore the putative Parvalbumin neurons in this brain area, it seems like all of the PD GPE neurons, when we do our 30-second long light pulse, show increases in their firing rate, and this persists for the entire 30 seconds. But interestingly, if we look at the cells that aren't expressing Parvalbumin, we don't see that they're just kind of sitting there. We see that they're actively inhibited during our light stimulation. And we think that this is due to the actions of these inhibitory collaterals within the GPE. So when we turn just these parvalbumin neurons on, not only are we increasing their firing rate, but because they inhibit the neurons around them, we're actually turning off this other subpopulation of neurons within the GPE. And this kind of natural dynamic of the neural circuitry within the GPE is occluded when we put channelrhodopsin in all of the neurons. So global channelrhodopsin stimulation is kind of blocking the turning off of this other subset of GPE cells. So who are these GPE neurons? Peter McGill is not going to like this slide, but in order to try to target these non-parvalbumin cells in the GPE, we used LHX6 transgenic mouse line, and we put the inhibitory opsin ARCH in these LHX6 cells. And we wanted to ask, well, if turning off of these cells is important for the rescue, then what happens if we just turn these off directly? And sure enough, when we put ARCH into the LHX6 neurons, we see that we are also able to induce this persistent rescue. And again, just like the parvalbumin experiment, when we inhibit the LHX6 neurons, we also get this persistent rescue. And we recover the same kind of constellation of behaviors expressed in the mice, similar to what we see in naive mice, with the exception of rearing. In terms of the mechanism of why this rescue is so persistent, it's something that we're still very actively working on. But we have a couple of clues from some experiments that we've already done. And I just want to summarize those really briefly here before moving on to the second part of the talk. So if you remember at the beginning, I kind of introduced the findings that neurons in basal ganglia output nucleus, their firing becomes highly irregular in the Parkinsonian state. And there are some really good predictions that this irregularity of firing of these neurons are contributing directly to motor symptoms of the disease. So we wanted to ask, what effect are manipulations in the GPE having on the physiology of these basal ganglia output neurons? So the experimental confirmation here is we apply our optogenetic interventions in the GPE, and then we look at the physiological effect that has on neurons in the substantia nigra reticulata, this major GABAergic output nucleus of the basal ganglia. So if we just look at the effect that dopamine depletion has on these neurons, just like we would predict, they have a very regular firing pattern in normal, healthy dopamine intact animals, which becomes highly irregular in the dopamine depleted state. You can see that these neurons are sort of have these waxing and wanings in their firing rate. They fire these kind of tight bursts of action potentials. And this is a pathological feature that is observed across animal models of Parkinson's disease. When we apply our optogenetic stimulations in the GPE, what we see is that these pathological bursts kind of get gradually trained out with successive stimulations. So not after the first pulse, but somewhere around the fourth, fifth stimulation, we start to see this decrease in the frequency of these pathological bursts. This is especially true for the most bursty of the neurons within the SNR. So if we kind of average across all of the neurons where we were able to maintain the unit across the stimulation protocol, you can see that these bursts start to get kind of gradually reduced, and then they stay reduced even after we stop stimulating. And this kind of matches the time course of the behavioral recovery that we saw in the open field. And then if we kind of keep recording, so we sort of move our stimulating electrode around to sample different cells within the SNR over this several hours long post period, you can see that for as long as we can maintain these animals on the wheel, you can see that the pathological burst firing doesn't really come back after our stimulations. This is also true in animals where we inhibit the LHX6 neurons within the GPE. But we don't get rid of these bursts if we do global channelrhodopsin stimulation of all GPE neurons within of all GPE neurons simultaneously. So we think that somehow, if I just sort of jump to this slide, so Long-lasting motor recovery is achieved by interventions that kind of cause a transient imbalance in the firing rates of these GPE neurons. So if you can decrease the firing rate of these LHX6 cells relative to increasing the firing rate of these Parvalbumin neurons, this seems to be the magic formula that you need to induce this persistent rescue. For the sake of time, I'm not putting all of the other experiments that we did into this presentation, but we also tried kind of globally inhibiting all the GPE neurons, and we tried stimulating the LHX6 neurons and inhibiting the Parvalbumin neurons. None of these other things work. It has to be this way. You have to do an intervention that increases the firing of PV and decreases the firing of LHX6 neurons. And then I also showed that this behavioral rescue seems to correlate with a reduction in pathological firing patterns of basal ganglia output neurons. And specifically, it's only these cell type interventions in the GPE that yield long-term motor recovery that are able to also yield long-term decreases in this pathological burst firing of basal ganglia output neurons. Okay. So let's see, looks like, all right, I'm about halfway through the talk, so I think I'm doing okay with time. So the next question that we wanted to address is how can we leverage this discovery of these kind of therapeutic populations of cells within the GPE and apply that to current treatments for Parkinson's disease. So this is an experiment that was done by 2 graduate students in the lab, Teresa Spix and Shruti Nanavadekar. So we know that if we are able to apply an intervention that is cell type specific, and we've even kind of found the cell types and we found the way that you need to tweak them within the basal ganglia. But how can we do that in humans? So there's this kind of almost paradox in basic research science is we're getting better and better at targeting specific neural circuits or specific subpopulations of neurons within circuits as our tools get better and better to intervene. But in a lot of ways, this ends up taking us further away from the actual treatment of human disease, because in the clinic, there's still really only very gross tools that are available for manipulating neural function. An example is deep brain stimulation. So this is increasingly common treatment that's used in very severe cases of Parkinson's disease. It's applied very late in the disease because it's an invasive neurosurgery where a stimulating electrode is implanted into the basal ganglia, usually kind of somewhere within that indirect pathway. And then high frequency electrical stimulation is applied. And miraculously, this seems to work very well, even though it's a very coarse kind of treatment. It's able to alleviate motor symptoms. I mean, for some patients, it's basically a miracle cure. But even if a patient has had DBS for years, once the stimulator is turned off, the symptoms come right back. So we wondered, is there a way that we can use this invasive treatment? So you're already putting a stimulating electrode into the brain. Is there some way that we can tweak that pattern of electrical stimulation that can make DBS more cell type specific and therefore maybe extend its therapeutic benefits beyond what we're currently getting with current practices? So is there a way that we can kind of use our insights about the neural circuits of the basal ganglia, and especially within the GPE, in order to kind of bring us closer to a treatment that might be able to be applied rapidly to humans? So for these experiments, we wanted to ask if we apply electrical stimulation kind of in the vicinity of the subthalamic nucleus. This is the most common anatomical target for deep brain stimulation in humans. And the subthalamic nucleus also provides the main excitatory drive to the GPE. Is there some way that we can kind of pattern this electrical stimulation such that it will preferentially increase the activity of these Parvalbumin neurons and decrease the activity of these LHX6 neurons? So for these experiments, we kind of went back to basics, and we did a lot of this study in brain slices because it allows us to visualize these different populations of GPE neurons while we're applying our electrical stimulation. So this is a sagittal section of a mouse brain. Here's the GPE, which we record from. So we put a recording electrode in one of these neurons. And then here's our stimulating electrode. And then we can target either LHX6 or Parvalbumin neurons during these experiments. So when we first apply electrical stimulation, these GPE neurons fire spontaneously. So even if you block all synaptic transmission, they have intrinsic pacemaking capabilities so that the baseline firing rate of these GPE neurons is around 15 to 20 hertz. And then if you apply electrical stimulation, similar to what is used in deep brain stimulation, so this would be 100 hertz, very high frequency electrical stimulation. On average, this tends to slightly increase the firing rates of these GPE neurons. And it doesn't matter if they're Parvalbumin or LHX6, kind of continuous 100 hertz electrical stimulation causes a slight increase in firing of these GPE neurons. So I'm going to be showing a lot of these graphs. On the y-axis here, I'm plotting the neuron's baseline firing rate before any electrical stimulation was applied. And then along the x-axis, this is what we're calling a modulation factor. So if neurons increase their firing rate over baseline, they're going to show up on the right side of the graph. And if neurons are inhibited during stimulation, decreasing their firing rate, they'll show up on this side of the graph. So again, just with stimulation that approximates conventional deep brain stimulation, it kind of biases all these GPE neurons towards slightly higher firing rates. Okay, so DBS doesn't just magically give us cell type specificity. We kind of knew that going into this. So now it's kind of time for the basic science. So is there something about the synaptic transmission during electrical stimulation that we might be able to leverage in order to tweak this stimulation and give us some cell type specificity? So just to kind of survey what's going on and what these GPE neurons are hearing synaptically during electrical stimulation, we went ahead and did a voltage clamp experiment. So we can, while we're stimulating, we can kind of change the holding potential that we clamp these neurons at. It's almost like tuning your radio station to a frequency to listen to the excitatory input they're getting. And then in the same neuron, we can kind of change the holding potential or change the radio station and listen to the inhibitory inputs that these neurons are getting. So during electrical stimulation, they receive both excitatory and inhibitory inputs. Then if we crank up the electrical stimulation and look at what these neurons are hearing during conventional DBS, what we see is that the LHX6 neurons get this initial barrage of inhibition right as you turn electrical stimulation on, but then it very rapidly decays. So within 3 seconds of turning the stimulation on, this massive inhibitory current is already mostly decayed. In the Parvalbumin neurons, you can see that this inhibitory current is basically absent. But by contrast, there's a stronger excitatory drive onto these cells, especially right at the beginning when you just turn stimulation on. So when we were kind of originally looking at the firing responses of GPE neurons during conventional DBS, we were looking at stuff kind of like way out here. We were looking 30 seconds after stimulation had started. So we were looking at firing rates at a time where these kind of synaptic differences had already decayed. If we listen to the extracellular firing rates of these GPE neurons, so this was actually done before breaking in, you can see that this LHX6 neuron is firing, spontaneously, and then we turn on the stimulation. There's this huge inhibitory current that rapidly decays, and that corresponds to this pause in the firing of this neuron right at the onset of stimulation. But then as the inhibitory current decays, the firing comes back on and actually speeds up a little bit. In contrast, in this Parvalbumin neuron, right when we turn stimulation on, this big excitatory synaptic barrage causes an increase in the firing rate of the neuron kind of right at the onset of stimulation. And then the firing rate stays a little elevated over baseline, but mostly it kind of goes back to where it was before. So this suggested to us that, just based on the kind of synaptic asymmetries that these different types of GPE neurons receive, it gives us a little window, a very narrow window, but a window where we can apply our electrical stimulation and really kind of augment these natural synaptic differences within the STN to GPE neural circuit. And so we hypothesized if we just keep our stimulations brief and apply electrical stimulation as bursts of 1 second or less of stimulation, then we should be able to kind of reliably inhibit the LHX6 neurons and reliably excite the Parvalbumin neurons. And this is exactly what we saw. So applying DBS as a burst stimulus instead of just kind of a continuous stimulation, we were able to get robust excitation of the Parvalbumin neurons in the GPE, but inhibition of the LHX6 neurons there. So this now so kind of using what we learned about the synaptic composition of the GPE, we were able to convert a non-specific electrical stimulus into a cell type specific neuromodulatory stimulus. Let me see what the title of this slide is. I can't see it. There we go. So the last thing that we wanted to address before going in vivo and trying this electrical stimulation in vivo is what is the kind of minimal neural circuitry required to get this cell type specific effect. So, we were sort of going in blind. We just kind of put our electrical stimulations, our electrode, somewhere in this axon bundle from the STN to the GPE, and we might only be stimulating input from the STN, or we might be stimulating the axons of other things that are kind of passing through this bundle. And this is going to be really important for the application of this stimulation in vivo. So we wanted to understand, especially where this inhibition was coming from onto the LHX6 cells, because that was not really expected. And it seems to be quite critical in order to get this cell type specific modulation. So we kind of tested a bunch of different hypotheses to figure out like what is the minimal neural circuitry we need to get this cell type specificity. And the first thing we tested, and what I thought was the thing that was going on, is that we're getting kind of feed forward inhibition within the GPE. So excitatory drive from the STN is maybe kind of biased towards these Parvalbumin neurons. It gets their firing rate up, and then the inhibitory collaterals within the nucleus then inhibit the neighboring LHX6 neurons. So working similarly to how we applied the channelrhodopsin stimulation. So to test this hypothesis, we put channelrhodopsin in the STN and used optical stimulation instead of kind of global electrical stimulation. And but what we saw is that the STN, the excitatory drive from the STN, seems to similarly excite both populations of GPE neurons. So both LHX6 neurons and Parvalbumin cells are excited by input from the STN. We're not, just by stimulating STN input alone, we're not kind of able to recapitulate that cell type differences in responses that we saw with electrical stimulation. So, so that didn't seem to be right. That's not the only thing that's going on and the only thing we need to stimulate in order to get this cell type specificity. So the next idea is, well, maybe this inhibition of LHX6 neurons is coming from the striatum, and maybe the inhibition from the striatum preferentially inhibits the LHX6 neurons. So if you kind of look at the classical diagram of basal ganglia circuitry, the majority of inhibition to the GPE is coming from these indirect pathway medium spiny neurons that are enriched in D2 and A2A receptors. So for this experiment, we just put channelrhodopsin in all of the striatum, and we stimulated whatever striatal terminals were present in the GPE. And when we did this, we saw that all GPE neurons were inhibited by this manipulation. So, so STN stimulation excited all GPE neurons, global striatal stimulation inhibits all the GPE neurons. this is still not giving us this, very cell type specific response that we see with electrical stimulation. So this hypothesis doesn't seem to be right either. And at this point, we're really kind of scratching our heads. And we're like, well, where could this inhibition be coming from? And here's one of the things that really always kind of bothered me about this striatal experiment. So if you think about where the main input from the striatum to the GPE is coming from, it's, like I said, it's coming from these D2 or A2A expressing medium spiny neurons. Their axons go from the striatum to the GPE, but our stimulating electrode is positioned here, kind of behind the GPE. So even though this is a major inhibitory input to the GPE, it doesn't seem like one that we would be recruiting with our electrical stimulation. But instead, if you look at the path of axons from D1 expressing medium spiny neurons, so these direct pathway neurons, as they're headed towards the basal ganglia output nuclei, the GPI and the SNR, our stimulating electrode does sit like right in the axon path of these direct pathway neurons. But if you kind of look at the classical textbook drawings of basal ganglia circuitry, these D1 neurons aren't thought to project to the GPE. So this was, a little bit weird. But if you go back, there's some really elegant anatomical work that's been done, 40 years ago that shows that there actually are axon collaterals from direct pathway neurons that target the GPE. And there have even been studies recently, there have been about 3 or 4 studies that have come out just within the last 5 years that have sort of rediscovered these collaterals and show that they're actually, they can be quite powerful at modulating neural activity within the GPE. So our stimulating electrode is positioned in an area where we could be kind of antidromically activating these direct pathway neurons, and therefore their inhibitory inputs to the GPE. So we asked, could these direct pathway neurons, could their collaterals that they drop in the GPE, could they be selectively targeting these LHX6 neurons and therefore be the source of inhibition? And that's exactly what we found. So if we put chanorodopsin only in the direct pathway neurons, we see that we can reliably inhibit the LHX6 neurons in the GPE, but there's very little effect on the firing rates of these Parvalbumin neurons. And that's shown across the population here. So putting this all together, these experiments told us that the minimal circuitry required to get these cell type specific responses using electrical stimulation is that you have to position the stimulating electrode somewhere that you can recruit this excitatory input from the subthalamic nucleus, but also this inhibitory, they like target these, antidromically stimulate these direct pathway medium spiny neurons. And this inhibition is kind of selective for the LHX6 cells. So if we were to put our stimulating electrode in the subthalamic nucleus, which is the major target for stimulating electrodes in human Parkinson's patients, we might not recruit antidromic stimulation of these direct pathway neurons. But instead, if we put our stimulating electrode more in the vicinity of the GPI, which is called the entopeduncular nucleus in mice because it's so tiny, then this is perhaps a better position for the stimulating electrode because we can get inputs from both the STN as well as antidromically stimulate this inhibition from the direct pathway neurons. So we went ahead and we tried this in vivo. So we, it ends up being actually very hard to do DBS in mice because they're very small and they can't stand to have a lot of weight on their head. So we went out to UCSF to learn how to do this deep brain stimulation from John Shore, who was a graduate student at the time in Alexander Nelson's lab. And so we made these homemade stimulating electrodes, which is kind of a series of twisted wires. And then it gives us 3 different electrical contacts that we can stimulate across and just sort of sample until you find a stimulating, like a pair of electrodes that give you a motor response. And then we kind of apply our stimulation that way. So the design of these experiments is it was a crossover study design with a one-day washout. So half of our mice got conventional DBS on day 1 and then burst DBS on day 2, and the other half of the mice had these treatments applied in reverse order. So burst DBS on day 1 and conventional on day 2. But the critical thing is that all mice got both kinds of stimulation. So we stimulate either in conventional mode for about 30 minutes or in burst mode for about 30 minutes. And the results were really remarkable. So if mice received conventional DBS, during the stimulation, they were able to start moving around. So we saw a decrease in the immobility that they have in the open field. But as soon as we turned the stimulation off, the immobility came right back. In contrast, the day that mice received the burst DBS, we did see that during the stimulation, they were also able to start moving around. But really excitingly, after we turned the stimulation off, the mice were able to maintain that restored movement. And for the duration of the experiment, they continued to move around the arena. This is the same data kind of just plotted in a different way. So this is the percentage of mice in our cohort that were able to remain moving. And again, it's the same mice. So when mice got conventional stimulation, you can see that immobility pretty rapidly returned. The therapeutic effects rapidly decayed is kind of the other way of interpreting this graph. Whereas mice that received burst stimulation, there were some mice that there were maybe 2 or 3 mice in our cohort that weren't able to maintain moving for the 3 hours after stimulation. But by and large, many of the mice remained highly mobile after stimulation. And then I just want to end here with a movie. So this is the same animal on 2 different days, receiving either conventional stimulation or burst DBS. So here they are during the stimulation. And then right after the stimulation, there are some carryover effects in both conditions. And now 1 hour after applying the stimulation, you can see that the mouse is completely immobile again after conventional DBS, but is continuing to move around after burst DBS. This just shows that they can kind of stop and change direction. So in conclusion, I've shown you that coincident excitation and inhibition of neuronal subpopulations in the GPE induces a long-lasting motor rescue in a reversal of Parkinson's associated pathophysiology in the basal ganglia output. And then we used this kind of foundational knowledge about cell types within that we made using optogenetics. We used this information to tweak the way that electrical DBS is delivered. So we were able to tailor our stimulation protocol so that using bursts of stimulation, we could drive population-specific neuromodulation within the GPE. And when we applied this as DBS in vivo, we were able to provide long-lasting therapeutic effects. So we're sort of calling this a circuit-inspired deep brain stimulation. With that, I'd like to thank the members of my lab that did this work, including really heroically doing this during the pandemic. And I didn't have time to talk about some of the kind of modeling work that we did to really explore the extent of parameters that give us this long-lasting rescue, but this was done by some of our collaborators at Carnegie Mellon in the computer science department. So with that, I'd like to thank you all for your time and attention, especially I know it's hard with all of these Zoom seminars, and I will take any questions.
Thanks so much for a fabulous talk. If anyone would like to ask a question, pop your hands up. I see one, Julian.
Yes, hello. I'm gonna jump in right now. So I work with Pete, and so I work on LHX6 Cree mice, and indeed the field is not really agreeing right now on like what should be the subdivision considered into the globus pallidus. But so it turns out like for us, LHX6 and PV are quite overlapping population, and it turns out one of your article as well in the Nature Neuroscience So you went from 2% overlap in the Journal of Neuroscience to 48% overlap in the Nature Neuroscience to 2% overlap in a science paper. So the only reason I can see would be like perhaps a discrepancy between LHX6 GFP and LHX6 Cree mice, maybe. And yeah, obviously, so it's very interesting the network effect you're describing, but obviously, so there is like almost a half of the GP subpopulation that you're not considering, which for us, like we described by FOXP2 marker. So I would be just curious to see what do you think they would fit into your model?
Yeah. Yeah. So yeah, I think for sure the LHX6 GFP line is more specific than the LHX6 Cree line. So for the more recent study with electrical stimulation, where we're just using a fluorescent marker to differentiate the cells, we get pretty good separation in terms of fluorescent markers between PV and LHX6 neurons, similar to what we kind of first characterized in our JNeurosci paper a while ago. The overlap really comes from the LHX6 Cree, because I think that expression is a lot stronger. In that Cree line, you also get amplification from viruses, viral delivery of chanorodopsin. So, you know, we've been doing a lot of work with intersectional strategies to try to minimize that overlap. And I can, you know, talk to you guys a little more about that offline. There's, we have some data that suggests that even if we use LHX6 only population, if we inhibit those, we can get the rescue. If we use the PV only population, excite those, we get the rescue. So it does look like, even though there is some overlap, that's not enough to kind of obscure a result that's really there. In terms of, you know, the archipelagal population, we're interested in those. I feel like there's lots of people studying those. So I'm sort of hoping that somebody else in the field will look at their role in this persistent rescue. You know, we've been doing some experiments with the NPAS line, but they not only target archipelagal cells, they also target a lot of neurons that project downstream and look similar to the LHX6 only population. So that mouse line is a little complicated, and we have the FOXP2 line. We haven't done tons with it yet, but we're having a little trouble getting super reliable expression in that line. There's also a lot of spill into the striatum whenever we use it. So I don't have any answers for you in terms of how that archipelagal population is involved in this rescue. We've certainly thought a lot about it. And, you know, if you guys want to do the experiment, that would be wonderful. Please let me know how it turns out.
All right, if anyone else has a question, pop your hand up. Pete.
Thanks, Lauren. Sorry, I was just wrestling with the screen there. Aaron, I wondered if I might ask you a really dorky, detailed question, and then if I'm allowed by Lauren to ask you a philosophical one. Could we start with some dorky stuff first? So,
I love dorky stuff.
All right. So I was a little bit confused, and it could be all me, by the segue. If you could go to your slide where you showed your initial experiments ex vivo, stimulating kind of just in front of the STN, and you said when you use conventional DBS, you see a slight increase in just about everything, right? That one there. So how do we go from a sort of, I see a bunch of excitation, to then when you started looking at cell types in the next slide, you start to see the inhibition within LHX6. How do I marry those together?
Yeah, let me see. I don't think I have the slide. So this is an example of one neuron. In the paper, we show maybe like 4 representative neurons. And in some neurons, you do see like a super brief pause here that when you show it over an expanded scale, kind of gets overshadowed and you don't notice it. So for this experiment, we averaged the firing rate over 30 seconds. And even the LHX6 neurons show a speed up, even though if we were to take the same neuron and just average over the first second, you would see that they're inhibited. So, so what happens is these LHX6 neurons, and I mean, you can kind of see this here. although perhaps not super well. Is that blurry? It's blurry on my screen. There it goes. So if we were to, and this only shows 3 seconds. So if we were to average the firing rate of this cell over 3 seconds, you'd see maybe a very slight decrease, but not much. But if you, center the window over like 1 second, then you see that it's turned off.
I see. So I see. Oh, okay. And I think the finding that antidromic activation of the D1 expressing direct spiny neurons is really interesting.
Mm-hmm.
But what I was struck by was you didn't mention at all antidromic activation of the GPE neurons themselves, which I would expect a lot of if you stimulate in and around the STM. We think all of those neurons send their axon through there, right?
Right.
So I'm sort of, maybe you could just comment on how often you saw antidromic activation. And if you didn't see it much, it could be because of you've cut the slice.
Mm-hmm.
And I would maybe expect much more of it in vivo, and maybe what role that might have to play.
Yeah. So I think, a detail that I kind of glossed over is that the stimulation that we're using is relatively, like, the stimulation amplitude intensity is relatively low. And, it seems like it's enough to antidromically act. Well, we can kind of geek out about this a lot more when we talk later, but I think that, like, the excitability of axons from GPE versus striatum, they're different in ways that we don't understand very well yet, because the experiments are, like, very hard to do unless we get, like, voltage sensitive dyes or something. And another thing that I will just, so we see antidromic stimulation in maybe 15% of the GPE neurons, but it's not an overwhelming majority. And I think that if we were to crank up our stimulus intensity, we would see a lot more. And so there's also kind of this window in stimulus intensity, like, you don't want to stimulate too hard, or you probably also lose the cell type specificity, because you'd be antidromically stimulating everything, just like you,
I see.
just like you're suggesting. I will also just show this slide. Sorry, it's so small, but I'm sure you're very familiar with what the STN looks like. This is an anatomy experiment using our intersectional tools, and it looks like the LHX6 only population, because we're using, like, a flip Cree intersectional system. So we're turning off expression in any cell that is PB flip. So that's how we think we're getting this LHX only population. They're kind of targeting this, like, very medial territory of the STN, whereas the Parvalbumin only population is targeting the more medial or central territory of the STN. Sorry, not medial, central territory. So and all of this is slightly moot, because it turns out that putting your stimulating electrode in the middle of the STN might not be the best place to target anyway. But probably, like, the site that neurosurgeons typically target would be the part of the STN that is getting kind of comparatively more innervation from the Parvalbumin neurons. So if you're going to antidromically activate something, it would probably be those, which would work in your favor anyway.
Okay, so the philosophical question is, and you said a really key word, and that's neurosurgeon. So the key part in the translational chain, right, is to get a medically qualified individual in a really expensive place to do this on humans. So Lauren, maybe you could, Aaron, maybe you could just comment on maybe your successes or the challenges and trials and tribulations in taking a basic discovery finding that's a really clear result. And how do you change the mind of neurosurgeons to drop what they've been doing for 20 years and try something new?
Yeah, I mean, so it's I've been talking a lot with neurosurgeons. It's been very interesting. So neurosurgeons, I find, are incredibly eager and willing to try new things. And the problems are all of the regulatory bodies surrounding them and what they have to, like, work in. One of the things that we really have going for us is that the stimulus parameters that we think would kind of maximize the therapeutic duration fall within FDA, or at least for America, like, fall within FDA approved stimulation parameters. So all you have to do is get them to run it in burst mode instead of just, like, leaving it continuously on. And that's something that neurosurgeons and patients are like very willing to try. But you still have to, anyway, so we've been talking with a group of neurosurgeons in Pittsburgh. I mean, we've had these, this has been a story that's, you know, things always come out years after the work is done. So, you know, we've been talking with them for the last 2 or 3 years, and we've been talking with device companies, and finally all of the pieces are together that the neurosurgeons in Pittsburgh are going to try they're going to start with a tolerability study, which hopefully everything's in place now. So hopefully that will happen within the next couple of months. Right now they're recruiting patients for that. But then going from that to like an actual clinical trial of if we turn this to, you know, burst mode, then how long do the symptoms last? That's going to be a little longer, but it is something that's actually relatively easy to implement clinically because the parameters aren't something crazy. The patients don't also have to be on some sort of drug, or it's like treatment that they would probably have anyway. If the burst mode is not tolerable for them, then you just turn it off and go back to conventional mode. So there's a pretty low bar to entry, which I think is something working in our favor.
Thanks so much.
Yep.
I guess I have a question as well to kind of piggyback on that. So you were saying at the beginning of the talk that with the conventional therapy, there is a loss of efficacy over time. Is that correct?
Yeah, yeah.
And so is your thought that if rather than using the conventional mode, you are more selectively using the stim and you don't then have to stim as frequently, that you think that you'll just, I guess, like, do you think that the loss of efficacy is kind of like a physical thing, like the stim electrode causing kind of a lesion almost, or like is it just that the cells stop responding?
Yeah. You know, I don't have a great answer to that question because how conventional DBS works is still, you know, like it's really kind of not clear. And I also, you know, from talking with neurosurgeons, get very different answers about how stable DBS is over time or not. Some of them are like, yeah, I've had patients that have had it for 20 years and they're fine. And some of them are like, you know, we need to bring them in for retuning of the stimulation parameters every couple of months. And then like some of them don't like it. And so it's super hard for me to actually get my finger on the pulse of the clinical community to know what are really the challenges. But, you know, my kind of my impression of where the clinical field is in terms of DBS is that for some patients, you get it in the right spot, it works perfectly, they're fine. But for a lot of patients, you know, for some patients, you never find the right spot, like the side effects are so strong that they can't ever get DBS. You know, for other patients, it's okay, but for whatever reason, either because of disease progression or because you're driving plasticity by constantly stimulating the brain for years, like you have to be continuously changing how you're stimulating to maintain the therapeutic effects. So how I envision that this could impact how we apply DBS is maybe patients just need to get, you know, a 30-minute bout of stimulation 2 or 3 times a day. And then, so that would greatly reduce how much stimulation they need. It would prolong battery life. It would hopefully reduce the accumulation of side effects because you wouldn't need to be stimulating for that long. And then, you know, one other thing that was, I think, kind of embedded in your question is, you know, when you stimulate either conventional or in burst mode, what kind of plasticity are you inducing? And, you know, that's something we're very interested in exploring. We don't yet know if you apply burst stimulation for a prolonged period of time, is that going to induce plasticity? And would that be good plasticity or would that be bad plasticity that would make it not work as well? We don't know yet, but we'd really like to answer that question.
Cool. Any other questions? All right, I think with that, thank you so much, Erin. I guess we'll give you a little bit of a break and then see you in some meetings soon.
All right, awesome. Thank you so much.
See you soon.
Bye, see you soon.
Bye, see you soon. All right, I'll stop recording.